Yeah, so I'm going to talk about the organoid tissue modeling shared resource. So we're a new shared resource on campus, and I suppose we term ourselves more an emerging shared resource. And just to come on. So here is the way we're set up at the moment. So I co-direct this with Bruce Apple, and we have a lab manager, Sean McGrath, who has expertise primarily in IPS technologies and gene editing and organoid technologies. And then we have 3 PRAs, Monica, Brandon, and Cole. So Brandon and Cole are relatively new, and Monica is just about to leave us to go to graduate school. So what are the goals of OTMSR? So we really want to increase the types of in vitro models that we have on campus, especially for human disease and development, and trying to develop protocols to model diseases and new strategies for drug therapy. So what we try to do is to provide training and protocols for all of these sort of high-end technologies to be able to generate organoids and actually establish organoid libraries. And we also provide authenticated and subsidized reagents. So we're trying to create a sort of cost reduction for people. And lastly, we have our education and training. We have in the past run a 3 weeks CSD organoid graduate course, and we're going to start that again, but we obviously, because it's a hands-on course, we got delayed because of COVID. We also do hands-on training within the shared resource, and in the future we'll be having workshops and symposia. So because we are a new resource, I thought I'd just tell you how we developed over time. So originally, I ran the enteroid stem cell core through the Gallup transformational grant that Sean Colgan had, and that started in July 2016. And then Bruce and I, we established the organoid and tissue modeling program in late 2018. And then in January of 2019, we got support from the Cancer Center for 3 years. And then we also applied for CERC funding to obtain an imaging 3D imaging system that we have in the facility. And then in spring 2020, the Diabetes Research Center P30 grant was funded, and we support the disease modeling core facility there. And then in January 21 was a busy year. We actually moved to new space, which was fantastic. And so this is a shared space between my lab and the shared resource. That's when we hired Sean, and we also got funding from the Gates Center for another 5 years for financial support. We actually started our website in July 21 and iLabs in 21. And then in August 21, we hired Cole and Brandon. And that's when we also started partnering with Steve to provide organoid technologies for the cell technology shared resource as well. And then in 2022, obviously, the Cancer Center P30 has been funded, and we've also submitted digestive disease P30 with Sean Colgan that will actually hopefully replace the transformational grant. So what does that look like in terms of how we perceive OTMSR on campus? So at the moment we have the Gallup, which is basically running out. We've had that initial support from the Cancer Center, and we have this ongoing support from the section of developmental biology through Bruce Apple and the Department of Pediatrics. So we want to be the umbrella program for all technologies on campus. And the idea is that we're trying to reduce redundancy of these services on campus, but also to have standardized protocols and subsidized reagents that we can provide. So what that looks like now is that we have the Diabetes P30 grant supporting us. The Gates Center is supporting us. The Cancer Center through Steve is supporting us through the cell technology shared resource. And then we have the pending digestive disease P30. So in terms of infrastructure, it's a little bit different. So we actually have, while we're trying to reduce the redundancy and have this centralized system, actually all of these entities have their own IRBs or EABs associated with them. So I'm not going to go into the details there. That's the complicated part. But the key for the investigators that want to use the core is that you get a 15% subsidy if you're actually a diabetes center member and a cancer center. So you'd actually get a 30% subsidy on your services. So what are organoids? So these are self-organizing structures, and they really come from stem cells, and they try to replicate sort of the in vivo architecture and differentiation into different cell types. And so there are two types of organoids. These are pluripotent stem cells and organoid adult specific organoids as well. And these are usually grown either in a 3D matrix or in suspension. And in most cases now, we sort of perceive these organoid systems to more closely reproduce or represent in vivo tissues. So what's the difference between pluripotent stem cells and adult stem cells? So the pluripotent stem cells, they really recapitulate development. They're fetal-like in their differentiation, and they involve very complicated directed differentiation protocols, very meticulous tissue culture maintenance, and usually they have an endpoint. So you do the differentiation and you get to the differentiated organoid cell type that you want, and then you do the analysis. If you want to do the experiment again, you have to go back through the directed differentiation. Where the adult stem cells are a little bit different. They actually replicate injury repair mechanisms, so it's more a regenerative type state. These are mainly of epithelial origin. They're relatively easy to generate. They're highly expandable. They actually do retain genome stability and can be maintained long term. So in this case, they're more like cell lines, but sort of sophisticated cell lines. The problem with these are that they are actually mainly just epithelial of origin, and then you actually have to co-culture other cell types if you want to get cellular complexity. So these have been applied to lots of experimental and translational applications, and I'm not going to go through this. But because both IPS and adult organoids really have applications in all these types of areas, from basic research to even all the way to personalized medicine in the future. So what are our services for the adult stem cells? So we provide protocol development and organoid production. So this is both consultation and training, the protocols for cell isolation, organoid generation. We mainly are focused on mouse and human, but we can actually do lots of other different species if people are interested. And in general, it's more about published protocols that are well established. We're not really helping develop new protocols. That for us is more a research project, but we can provide all the reagents to help you do that if you're interested. And the important part that we do is we provide specialized reagents. So, and these are mainly because we buy them in bulk, they're subsidized as well. So batch tested growth factors, conditioned media, Wnt and organoids, and as I said, subsidized commercial reagents, Matrigel and growth factors, etc. And we could also provide you with complete organoid meter if you want us to make that for you. And obviously we have to authenticate, and this, I don't have to repeat, this is basically what Steve was saying before. We can provide organoid libraries, but we really have to have the validation, so SDR analysis, mycoplasmid testing, etc. And also we can store in cryostorage. Both organoid libraries and any types of organoid tissues that organoids that you would like. I'm actually finding the elevation a bit hard up here. So the other thing that we can do is assay development. And here I'm just showing you two parts of what we do for assays. So we do medium throughput real-time live and endpoint assays. So on the left here, I have a mantis, which is basically a fluidic handler that bioprints matrigel organoid suspension. So we can accurately add very small amounts of these suspensions to any type of plate format that you're interested in. And on the right here, we have the imaging system that we have, which is more unlike the IncuSite. This is more like specific individual organoid analysis, more at high end. So it complements what Steve's got in terms of the IncuSite organoid modules in the CTSR. And we can also help with protocols for growing these three-dimensional structures in 2D on transwells and looking at other protocols in terms of metabolite and pathogen interactions. So how do you grow organoids? And I thought it was good just to describe that. So this is from the first example of this from the Cleavers lab. And basically the idea is the reason you can grow organoids is that you're recapitulating the stem cell niche in vivo. And so here's just in the small intestine, which was the first example published, where the niche factors, the positive niche factors are Wnt, R-spondin, and EGF, and then there's a negative gradient of BMP signaling. So you add a BMP antagonist, noggin. And so the original culture conditions was just adding R-spondin, noggin, and EGF, and these stem cells would grow into organoids. And then 2 years later, they established another protocol for human intestinal organoids. And you can see it's a much richer media, but it still has this essential Wnt, R-spondin, EGF, noggin type component to it. And then just last year, they generated another protocol for thyroid organoids. This is probably the most recent one. But again, it's on the same theme of supplying these different types of niche factors. And if you look at what that protocol has been used for, basically nearly every epithelia that we have in the body can now be grown as organoid structures. And the ones in orange are the ones that we've actually done on campus to date. But most of these have established protocols, and we're quite willing to help people establish organoids from those different tissue types. So of course, the key question here is why do we do this? And one of the drivers was because a lot of these factors are extremely expensive, and they can be actually relatively unstable. So recombinant Wnt's renowned to be unstable in culture. And the commercial medias are available, but they're extremely expensive. So 100 mils cost you $500. And these are proprietary formulations. So it's very difficult to use these because we can't find out what's actually in those medias. So by generating our own condition media, we can actually manipulate the culture conditions and have it defined. So this is just showing on the right our clean tissue culture suite, which is where we generate the condition media, and this is where we do our IPS work. And here you can see our bioreactor system that we generate this condition media. And I should say that I'm excited with our interactions with Steve, because this is something that we want to expand with CTSR on a number of levels. So I just sort of give you an example of what we do in terms of adult organoids. And this is work with Todd Pitts and Wells, Mr. Smith. And this is their, primarily their PDAC tumor xenograft system. So this pancreatic cancers. And we also have generated organoids directly from pancreatic cancer tissue as well. And this is all done in our dirty room. And as Steve was alluding to, you know, we are very concerned about mycoplasma from primary tissues, and that's why we have the systems completely separated. And here we have just our organoid biobanking system. And again, this is an example where I think Steve's going to be able to help us greatly as well as we move forward with his much larger storage system. So when we generate the organoids, we actually try to do analysis of the exact, we bisect the tissue if we can, and we have validation of the tumor tissue, and then we do all this validation of organoids that we generate. And I would say, I'm not going to go through the list, but I would say we have about an 80% efficiency of growing tumor organoids from this system. And it's greatly expanded the number and diversity of organoid lines that are available. And this just looks at a part of the panel that we have. I think this is 10 in this little library. And this is just showing what the original clinical mutational profile, the morphology of the organoids and their growth rates. And I'm just going to give you an example of the two of the assays that we use in the core. So this is where we're looking at a pancreatic tumor organoid line, and we're looking at a morphology-based assay where we're withdrawing different niche factors to see if the tumor will grow independently of these stem cell niche factors. And this is just looking at the morphology as we go and reduce the number of factors that are in the culture media. And so you couldn't do this with the commercial types of media. And you can see that this organoid line grows quite well with just R-spondin alone. And then we do convert the morphology-based assay, and then we do an endpoint cell type glow assay, and you can see R-spondin alone. And then when you withdraw R-spondin, the organoids stop growing. So this suggested that that this organoid line might have a role for Wnt autocrine signaling because of this R-spondin dependency. So just to look at that more specifically, we use the porcupine inhibitor to block Wnt secretion, and we use two different inhibitors, C59 and ETC159. And you can see both morphology-based and in the cell type glow, you just completely block growth. So this suggested that this organoid line is actually Wnt dependent. And so here I'm just showing you a description of what people have done and what we've done with this system. And so here you have an ability, looking at the biological phenotypes, you can define this Wnt dependency, which you wouldn't have been able to predict by genomic profiling. And Todd Pitts submitted some of these for bulk RNA-seq, both the PDX tumors and the organoids, and Andrew Goodspeed helped us analyze that data. And so when we actually looked at this PANC269, you could actually see that it actually has a Wnt-dependent signature, both in the organoid line and in the PDX tumor as well. So this is an example of how we can help people with their organoid systems. So just going on to the pluripotent stem cell derived organoid services and resources, we provide authenticated IPS lines for organoid production. And we can also help if people have got the specific IPS lines and want to help, we can help with directed differentiation models as well. Again, we provide specialized reagents, subsidized bulk reagents for these types of cultures. We're also developing gene expression and gene editing, including CRISPR in the system, and I'll get into more information about why we're doing that. And in the future, we're very interested in optimizing primarily orthotopic transplantation and also some live imaging with these organoids. So both the iPS-derived organoids, they actually can differentiate more if you transplant them under the kidney capsule. And that's one thing that we want to do. But if you can transplant it orthotopically into the tissue of origin, they may actually differentiate even more. So we actually have generated 4 of our own control IPS lines. There's a lot of issues with NTAs with IPS lines, so we've decided to develop our own. And these, have spread with different sex, age, and race. So it has a good representation that can be used for different experiments. These will be fully characterized karyotype pluripotency fingerprinting and mycoplasma analysis. And we're at the point now with these lines really testing their ability to differentiate into different cell types. And we're really focused on endodermal lineages initially. So this is just an example of one of those lines differentiated into human intestinal organoids. And you can see this process is quite a long process in terms of weeks to get there. And it's a fairly complicated, meticulous set of changes in growth factors to develop this system. So a lot of our investigators that are interested in this want to optimize these types of directed differentiations. Then none of them are perfect at the moment. So a lot of people are trying to use different reporters for different transcription factors or manipulating different genes to try to optimize these conditions. And that gets to our gene editing, because you have to combine a lot of these reporters with the sophisticated directed differentiation to get the analysis. So we decided that we needed to provide these services as well. And Sean McGrath's an expert in this area. So we're really mainly interested in fluorescent reporters at the moment, but we can also do SNPs or corrections or knockouts. And in the future, we would actually like to think that we can maybe complement with FGSR in terms of CRISPR screens in the future. So this is one of the goals that we have in the gene editing services, to be able to track multi-gene expression in IPS lines live. And so this is not live, these are whole mount images, but this is the type of area that we think is going to be very powerful as we move forward. And Sean McGrath's developed a whole series of fluorescent targeting plasmids that allow us to target specific genes with a T2A that has downstream of that a specific reporter. So we have this whole rainbow range of different fluorescent probes, both cytoplasmic and nuclear, that can be used for these studies. And this is just examples, some of the ones that we've done already in terms of in IPS lines. So FOXA2 and Brachyury are both nuclear reporters. Okay, so that's sort of where we're at. We're still developing a lot of these things, but I think what I wanted to emphasize is that how we interact with all the other facilities on campus. And I might start over here. So I'll start with the drug discovery. Obviously, we generate a lot of organoids, and if you really want to do high content, high throughput screening, we can provide lines that are suitable for that, for the drug discovery shared resource. With the Gates stem cell biobank core, they're helping us with reprogramming to generate IPS lines. And as Steve mentioned, the molecular biology core, which has different names, but also called the disease modeling core, that's helping us with SDR analysis and sequencing, etc. In terms of pathology, that's really essential for us in terms of our validation and authentication of what we're making, both histology, karyotyping, and for a lot of the gene editing, we're also using the droplet PCR machine that's available through SR. And in orange here is things that we're really interested in further developing. And for us, the pipeline is so important in terms of human tissues and tissue procurement. And Audrey has been helping us with the donor alliance program, where hopefully we can get a lot of normal and potentially other disease tissues from donor patients. And I think this is a great paradigm for us to move forward in terms of getting a nice pipeline towards getting human tissues. And the other thing that I would like to influence with Audrey is also sort of viable biobanking of samples as we move forward. BBSR, obviously Andrew and his crew have been helping us with a lot of the analysis, both bulk RNA-seq and whole exome sequencing. And then genomics, obviously, with sequencing itself. Flow is essential for us in terms of organoid purification, back sorting specific cell types, reporters. And I'll move up to functional genomics. We haven't really been integrated with functional genomics as yet, but lentiviral production, lentiviral shRNA libraries, we're starting to use these in mouse systems, mouse organoid systems, but obviously CRISPR screening in organoid systems, I think, is a future direction that we'd like to integrate. And obviously for today's talk, with cell technology, shared resource, and Steve, the IncuSite modules are essential for us in terms of investigators who generate organoids, or we help them generate organoids. They've got to be able to screen them. So this is a great resource, the organoid modules. I think the cryo storage in Steve's facility is also great for us in terms of backup. But the most important thing is when we move forward is just the ability that Steve has in terms of conditioned media purification and the FPLC that he has. And so in the field, we're moving away from standard expressed Wints, where we do have an Affiman Wnt3A expressing cell line. So this is a line that allows you to purify Wnt. Affiman is the stabilizing binding protein, and you can actually purify this away from serum. And so you can actually grow a number of organoids don't like growing in serum. So this is another way to enhance the types of organoids that we can grow. And then the next generation surrogate Wints, these are synthetic Wints that are expressed in like 293 cells. So this is going to be a fantastic resource for us. And then last, same slide as Steve in a way. I put CTSR here and us over here. And we're on the third floor of Barbara Davis Center, similar sort of scheduling through iLabs. We're generally Monday to Friday. Here's our email contacts and here's our website. And I'm happy to answer any questions.
Hi, Peter. This is Jill Slansky. I was wondering if you guys have done any addition of immune cells to your organoids.
So that's certainly I had HUMSA on my list as a future direction. I think I'm at a Keystone Organoid meeting this week, and a number of examples of co-culturing, purifying TILs, co-culturing immune cells. That's something we can do for sure. And I think in terms of CAR T cell, the organoids also can be, we can make them to express certain antigens, or they may already have them. So we may actually have validated models to test CAR T cell reactivity as well.
So for example, TGF beta, I imagine is everywhere in a tumor, but it's immunosuppressive for T cells, and so is there? I guess the timing of when the TGF beta is it always?
Yeah, no, that's a good. I asked someone this question last night. So I was talking to the Hubrick group, the Cleavers Hub Organoid group. So they had this service generating T cells from their tumor tissue, and so they're actually they're able to combine the media that we need for the organoid generation and the media for the immune cells, and then not having issues. So there's ways to do it. Yeah.
Oh, nice.
Yeah.
If you want to, I'd be happy to work with you on that. We have a mouse setup that we've been using forever, so we can provide.
Yeah.
Provide part of it, and you provide part of it.
Yeah. No. Yeah. That's. I suppose that's why we have this presentation. Yeah.
Thank you.
Sure.